Hello everyone. So, the topic for today's discussion is about development of the eye. So, the eye is the organ of vision. It is divided into two components, the eyeball and the accessory structures of the eyeball. So, the development begins at about the 22nd day of embryonic life and it develops from four different sources. They includes the neuroectoderm of the forebrain, the surface ectoderm of the head, the mesoderm between these two layers and the neural crest cells. So, the neuroectoderm of the forebrain forms the retina, posterior layers of iris and the optic nerve. The surface ectoderm of the head forms the lens of the eye and corneal epithelium. The mesoderm between the neuroectoderm of the forebrain and surface ectoderm of the head forms the fibrous and vascular coats of the eye, and the neural crest cells migrate into mesenchyme from the neural crest and form the choroid, sclera and corneal epithelium. So, the development of the eyeball can be described under the two different headings. One is the optic cup and the lens vesicle and the other parts of the eyeball. So, how does the optic cup and the lens vesicle develops? At first, a pair of shallow groove or the sulcus appears on either side in the forebrain. It is called the optic groove or the optic sulcus. So, the optic sulcus invaginates the surrounding mesenchyme to form the optic vesicle. So, each vesicle grows laterally and its distal part expands and invaginates to form the optic cup. Its proximal part connected to the forebrain constricts to form the optic stalk. So, here we can see the optic vesicle close to the forebrain or the diencephalon process. So, the optic cup is the distal part of the optic vesicle, whereas the optic stalk is the constricted part of the optic vesicle. So, the development of the eye is influenced by PAX6. It is the key regulatory gene for the eye development. So, the key steps are the formation of the optic vesicle and the optic stalk, which are the neuroectodermal in origin. So, the lens plecoid, we have seen the surface ectoderm which is close to the optic vesicle. It will become thickened and forms the lens plecoid. So, this lens plecoid get depressed below the surface to form the lens pit. As the lens pit deepens, its edges approach each other and fuse to form the spherical lens vesicle. Then it is cut off from the surface ectoderm. As the lens vesicle is being formed, the optic vesicle begins to invaginate to form the double layered optic cup. So, the margins of the optic cup grow and enclose the lens vesicle, that is, the lens vesicle enters into the optic cup. So, subsequently, a fissure called the retinal fissure or the choroidal fissure develops on the inferior aspect of the optic cup and the optic stalk. The vascular mesoderm enters into the choroidal fissure and gets trapped in the optic vesicle and optic stalk. Then the choroid fissure is closed. This vascular mesoderm within it forms the hyaloid vessels, that includes the hyaloid artery and the hyaloid vein that supplies the layers of the optic cup, mesoderm in the optic cup and the lens vesicle. So, now the development of other parts of the eyeball. First, we will discuss about the retina. The retina develops from the walls of the optic cup. So, we have seen the optic vesicle gives rise to the optic cup, right. So, the retina develops from this optic cup. The inner thick layer forms the neural layer of the retina, and the outer thin layer forms the pigment layer of the retina. The cells of the neural layer proliferate to form the various cells of the retina. So, that includes the photoreceptor cells, which are rods and cones, the bipolar cells and the ganglionic cells. So, the rods and cones are modified neurons. The axons of the ganglionic cells form the nerve fiber layer, which passes through the optic stalk and forms the optic nerve. So, during embryonic and early fetal periods, the pigment and neural layers of the retina are separated from each other by the intraretinal space, that is representing the original of the space between the inner and the outer layers of the optic cup. So, before birth, the space between the inner and the outer layers of the optic cup is obliterated with the proliferation of the cells of inner layer. As a result, the rod and cones comes in contact with the pigment cell layer of the retina. So, here we can see the outer layer of the optic cup that is going to give rise to the pigment epithelium or the pigment cell layer, and the inner layer of the optic cup, that is light sensitive neural layer, is derived from this inner layer. The optic nerve. So, already we have seen the optic nerve, the optic stalk contains the choroid fissure, which contains the hyaloid vessels. So, the hyaloid vessels later becomes the central artery and the vein of the retina. So, here we can see at the center of the optic nerve, the hyaloid vessels are present. So, the hyaloid vessels gives rise to the central artery and vein of the retina. So, the choroid fissure closes in 7th week. As a result, the optic stalk along with the axons of the ganglion cells forms the optic nerve. So, here we can observe the hyaloid vessels. So, the optic nerve is a tract of diencephalon and not a peripheral nerve, because it is myelinated by the oligodendrocytes in the 3rd month, and it is not capable of regeneration after transaction, and it is surrounded by the meninges and the subdural space filled with the cerebrospinal fluid. Now, the development of the lens. The lens develops from the lens vesicle. So, that is derived from the surface ectoderm, that is in close contact with the optic vesicle. So, initially the lens vesicle is lined by a single layer of the cuboidal cells. The cells of the anterior wall of the vesicle remains cuboidal, whereas the cells of the posterior wall of the lens vesicles elongate and become columnar and extends towards the cavity of the lens vesicle. So, as a result, the cavity of the lens vesicle is obliterated. The elongated cells of posterior wall of lens vesicle further elongate considerably and lose their nuclei to form the highly transparent primary lens fibers. So, the lens grows because the new lens fibers, that is known as the secondary lens fibers, are added to it by the cells in the equatorial zone of the lens. So, the cells of the anterior layer persist as epithelium. So, the secondary lens fibers continue to form during childhood and the lens increases in its diameter. The primary lens fibers becomes old and harder and it will be moved towards the center of the lens. So, that will become the nucleus of the lens. So, the surrounding, the periphery, it will be soft and forms the cortex of the lens. So, this lens is formed during 7th week. So, here we can see the structure of the crystalline lens. Anteriorly we can make out the anterior capsule, just below that there is the epithelium, and next to that is the cortex, which will be having the newly formed fibers, and towards the center the old fibers will be present and it will be hard. And here we can see the Y-shaped structure of the fetal nuclear fiber. So, development of sclera and choroid. So, the mesoderm around the optic cup differentiates into two layers. The inner vascular layer forms the choroid and the outer fibrous layer forms the sclera. Anteriorly, the sclera is continuous with the substantia propria of the cornea, and posteriorly it is continuous with the dura surrounding the optic nerve. So, the choroid is continuous anteriorly on each side with the ciliary body, and posteriorly it is continuous with the pia and arachnoid around the optic nerve. So, we have seen that the sclera continues posteriorly as the dura mater, and whereas the choroid continues posteriorly as the pia and the arachnoid. Ciliary body, it is a wedge shaped extension of the choroid anteriorly. It is formed by the mesoderm around the anterior part of the optic cup. The ciliary muscle and the connective tissue of the ciliary body are derived from the optic cup, that is mesoderm surrounding the optic cup. The pigment epithelium lining the inner aspect of the ciliary body, including the ciliary processes, is derived from the outer layer of the optic cup. And the non-pigmented epithelial layer forms the inner layer of the optic cup. The development of the iris, it develops from the rim of the optic cup. So, the epithelium is derived from the both layers of the optic cup, and it is continuous posteriorly with the double layered epithelium of the ciliary body. The neuroectodermal cells of the optic cup give rise to muscles of the iris, that includes the sphincter and the dilator pupillae. And the vascular connective tissue, they are developed from the mesoderm. So, the anterior extension from the rim of the optic cup, which forms the iris, does not extend up to the center. As a result, almost a circular gap remains in the center of the iris and forms the pupil. So, development of cornea, it is induced by lens vesicle and derived from the following. The outer stratified squamous epithelium is derived from the surface ectoderm. The lamina propria is derived from the mesoderm, and the inner corneal epithelium is derived from the neural crust cells. So, now the anterior and the posterior chambers of the eye. The mesoderm located between the developing iris and the cornea splits to form the space called the anterior chamber. The mesoderm anterior to this space forms the substantia propria of the cornea and the mesothelium of the anterior chamber. The mesoderm located between the developing iris and the lens splits to form a space called the posterior chamber. So, the anterior segment of the eyeball is divided by the iris into anterior chamber and the posterior chamber. So, when the pupillary membrane disappears, then the pupil forms a communication between the anterior and the posterior chambers of the eye. So, the anterior and posterior chambers are filled by aqueous humor secreted by ciliary processes of the ciliary body. Then the vitreous body or the vitreous humor. So, at first the vitreous humor is formed by neural crust mesoderm present in the optic cup. It is called the primary vitreous humor. Later, it is replaced by the gelatinous secondary vitreous humor derived mainly from the inner layer of the optic cup and a little bit from the lens vesicle. So, the vitreous body is made up of the transparent gel like intracellular substances, and it contains the hyaloid artery that later obliterates to form the hyaloid canal of the adult eye. So, now we will observe what are the adult components of the eye. So, we have seen from the neuroectoderm the optic stalk and the optic cup is derived. From the optic stalk, the epithelium of iris and ciliary body and the optic nerve is developed, and from the optic cup the retina, dilator pupillae, the sphincter pupillae is developed, and from the surface ectoderm we are having the lens and the anterior epithelium of the cornea, and the derivatives from the mesoderm includes the sclera, substantia propria and the posterior epithelium of the cornea, choroid, ciliary body and stroma of the iris, the vitreous body, and along with that the central artery and vein of retina and the extraocular muscles. So, we will see now the period of conception and what are the milestones occurs accordingly. So, during 3rd week the optic groove appears. 4th week the optic pit develops into optic vesicle, the lens placard and the embryonic fissure develops. During 1st month the lens vesicle forms and hyaloid vessels develops. During 1.5 months there will be closure of embryonic fissure, the proliferation of neural retinal cells and the appearance of the eyelid folds and nasolacrimal ducts. 7th week there will be formation of embryonic nucleus of lens, the sclera begins to form, migration of neural crust to form corneal endothelium, stroma and iris stroma. During 3rd month rods and cones precursors of differentiation occurs, the anterior chamber appears and the fetal nucleus start to develop. And during 4th month formation of physiological optic disc cup and the Bowman's membrane appears. 5th month the eyelid separation begins. 6th month there will be differentiation of dilator pupillae muscle, the cones differentiate and the nasolacrimal system becomes patent. 7th month rods differentiates and the myelination of the optic nerve occurs. During 8th month the hyaloid vessels disappear, and during 9th month the pupillary membrane disappears, and after birth the macular region develops further. So, clinical aspects: anophthalmia, there is absence of an eye. It occurs when the optic vesicles fails to form. Then cyclopia, that is a single eye. It is severely uncommon anomaly of the face. There develops a single median eye. So, the cyclopia is transmitted by recessive inheritance. So, the two eyes may fuse to form a single eye. Microphthalmia, a small eye. Here the optic vesicle is small and there is a underdevelopment of the eye. The lens is usually not formed. The microphthalmia is usually associated with intrauterine infections from toxoplasma, rubella virus and cytomegalovirus, etc. Persistent pupillary membrane. Here the pupillary membrane may persist as a whole or in the form of few strands of connective tissue. It may obstruct the vision completely or partially. Coloboma of the iris. It is a congenital notch or the cleft on the inferior aspect of the iris. The coloboma means a part that is cut off or missing. So, that completes the development of the eye. Thank you.